Morning, students. This is for the 5th semester B Pharmacy students, the 4th unit. We are today's already we have I think you have come across with the first chapter using most of the biosynthetic pathway. So now it is necessary for you to know about the basic methods of extraction, isolation, characterization. As we have discussed in the class, it's very important for the students should know about what is the difference or what is the main importance of identification of a crude drug or a herbal drug, and after authentication, after getting the approval from the authorities, and then we have to start with the extraction, then isolating or isolation or separation, and then the compounds have to be characterized, identified. We need to be identified and to be structural elucidation. All this comes in the characterization. So anyhow, today we are going to discuss about various methods of extraction, and we will try to have some portion in it. And this is usually to show sometimes extracts are filled in the capsules and even given in the form of tablets. So extraction, separation, isolation of that is plant constituents plays a major role. So what's the extraction? We will start from the basic definitions, and it will be easy for you to write sometimes the essay questions if they ask for detail about extraction. In the extraction process, in the animal source or the plant source, the drug is treated with particular solvent. The solvent dissolves the medicinal active constituents itself, but the plant or animal tissues will not solubilize. So we have to select in such a way the solvent for extraction to isolate or to remove the active medicinal active principle. So that will dissolve or that will be dissolved by the solvent which you are using it. So that's the definition which we can write. Extraction is the process, or you can say, in which the separation of the soluble constituents. So solubility plays a major role while selecting a solvent system in the extraction. Soluble constituents occurs from insoluble substance, either solid or liquid, by process with a specific solvent. And extraction of the plant material in general, all plant material, first it is necessary for properly authenticated by means of authorities. So time may not be wasted or money may not be wasted by examining in between while the research is going on. The materials in doubtful origin will not occur. So before starting a procedure of a research or before starting of a extraction, it is necessary for you to authenticate it by the proper botanist of the plant species or animal zoological species have to be identified. The choice of extraction procedure depends on the nature of the plant and even the type of the plant, whether it is what's the part of the plant, what is the condition of the plant, is also the material condition also plays a major role. And the component which you are interested to isolate, whether it is an alkaloid or an or a phenolic compound or an aromatic compound. So these are some idea. It has a parameter, it has a significance, it has an impact to select a solvent system or a method of extraction. So there are many methods of extraction which we should understand. Dried material is normally powdered or comminuted or size reduced so that the surface area for the solvent system will be increased and extraction will be improved. Before extraction, it is needed for you to pulverize, needed for you to size reduction have to be done for dried material. Fresh plants like leaf, flowers can be homogenized or macerated with a solvent alcohol. Alcohol is also useful for stabilizing fresh leaves. Usually we are using. So solvents, when you are using, just a picture of chloroform I am showing, it's a non-polar solvents. Usually alcohol can be used, but not in all constituents it is not possible. So we can use water immiscible solvents commonly used like light petroleum ether for volatile oil, fixed oils, steroids. If you want to extract, they are very good solvents by ether or petroleum, light petroleum ether can also be used. Chloroform is very good preference for alkaloids and quinones. So like that, even for phenols, if you want even for example, when you want to extract alkaloids, organic bases, basification of the plant material is necessary to get a proper extraction if it is water immiscible solvent is used. If aromatic acids or phenols is the constituents which you are interested to extract are going to be extracted, acidification may be required. So method of extraction process of phytoconstituents, there are various methods which you might have come across in various other subjects also. I am just going to revise you and you can go in detail what you studied in pharmaceutics and all, you can go in detail for your knowledge and as well as so that you can write more. So first you can see infusions, decoction preparation, digestion, maceration, counter current agitation, counter current by means of using a current of flow of you can see baffles or running of a solvent system menstruants through the material, counter current extraction, supercritical fluid extraction with liquid carbon dioxide, hot continuous extraction, succination, percolation, ultrasound extraction, ultrasounds waves can also be used for extraction, steam distillation, microwave assisting extraction. So there are many techniques nowadays available. Some of the terms which is needed for you to remind what you studied previously, I'm just like menstruum means in extraction, solvent or mixture of solvent, one solvent or mixture of solvents used for extraction. They are called menstruum. Micella, solution with extracted substance. Rinsing, you might have used this word in the laboratory, rinsing. Dilution of extractive substance out of disintegrated cells. Laxative and leaching. Leaching, not it's lixate or laxative, leaching. Water is used as a solvent for extraction, then it is called leaching. Mark the residue left after extraction. Press the mark, squeeze the mark. Like that you might have studied when you are doing the extraction procedure by percolation. The desired constituents, the mark which is present after extraction. Infusion, we will discuss about infusion. You see, infusion, preparation, infusions by ginger infusions, pepper infusion, clove infusions, mentha infusions can be prepared. In the infusion process, the crude drug is left with solvent to soak as shown in the picture, then properly at room temperature. Soaked properly in the room temperature for a particular period of time with or without intermittent shaking. After the specific period, the filtration will follow, which will separate the plant debris with the mycella, that solvent with extraction, the main constituents, which just now we have seen the definition for mycella. Various times the plant material has settled down as shown in the picture, which has separated by decantation and can replace with other solvent. So this is decoction. You might heard the word of decoction. This is also one type of extraction method. Decoction. Take drug in a vessel, add 1000 ml of water and cover it. Boil for about 15 minutes. Cool it to room temperature. Strain the liquid and press the mark. Decoctions. Digestion. Digestion is modified form of maceration in which the temperature of the menstruum is kept heated at particular temperature at under pressure. The rise of the temperature and pressure help the menstruum to penetrate the drug and extract it completely. The apparatus used for the digestion is the digester, which is generally made up of metal. So you know very well, pressure, temperature, they have an impact over even. In low temperature, it's possible for you to extract, even to boil some liquid faster without thermolabile substance in a fast. It will get evaporated, it will get heated, so that easily you can, even that is one impact of using the pressure and temperature. In a metal chamber, digestion can be done. It's also a type of extraction. The drug is placed inside of it with menstruum, the solvent which you selected for extraction, under specified temperature and pressure, and then screw the cover with the help of nuts. Maceration, you know this name always. Extraction means maceration, and percolation is a common name which everybody knows. But you should differentiate, and different types you should write. That's why I have taken this one for explanation. In the process of maceration, the crude drug is immersed into the bulk of menstruum or solvent for at least 3 days, generally 3 to 7 days. During this period, the menstruum is agitated frequently. The menstruum and the container should be kept in the stoppered container. The mixture is then filtered or strained through net or sieves for filtration, separation of the mark and the measles or the menstruum after extraction. The maceration may be various types, like simple, modified, multiple, kinetic maceration, remaceration can be divided. So comparison, when you see, when you are using organs like bark, roots, simple maceration, unorganized drug like gum, resins in modified maceration, drug the medium menstruum shaken occasionally 3 to 7 days, the same thing. Here also 80% menstruum shaken because it is powdered and unorganized drug. And strain the liquid with a press. You can press the mark, but here it is not possible because it is sticky in nature. So decant the liquid in the mark and it's not possible to press the mark. Mix the liquid, obtain and clarify. Filter. This can be filter. One liquid pass through a menstruum. You can add the menstruum to wash it again because it is an unorganized drug. Mix the liquid, obtain, clarify, and filter one liquid and pass through remaining menstruum. Filter it not adjusted to volume. The filtrate which you obtain is not adjusted to the volume. But here you have to adjust to the volume. Here the mark is pressed because it contains considerable amount of macerate, because it's organized parts of the plant like leaf, root, stem and bark. Here the mark is gummy, and because it is usually exudates which is obtained like a gummy exudate, usually they are unorganized drug. So they cannot be obtained, they contain any macerate, so pressing of the mark is not necessary in this unorganized drugs when you are using for more modified maceration method. And multiple maceration, the maceration process is used for concentrated preparation where the entire menstruum is segregated into two parts. That is double maceration. 3 parts means triple maceration. Each menstruum are used individually for the maceration process. In this process, the drug menstruum ratio is low. Drug and the solvent system ratio is low, and the extraction is performed with less amount of menstruum. Kinetic maceration. Like the simple maceration, the kinetic maceration is proceeded into room temperature. But the single difference is that the material is under constant movement. The intensity and type of movement plays an important role in this maceration. Shaking or mechanical shaking or a magnetic shaking can be provided. Remaceration. In the remaceration process, part of the solvent is added to the drug. After filtration process, the residue is treated with the remaining solvent. That is remaceration. Again repeated. Uses of maceration. British Pharmacopoeia permits the preparation of tincture by maceration method of drug, like squill tincture, catechu tinctures, senna liquid extract, compound benzoin tincture and opium tincture. Mother tinctures of most of the homeopathic pharmacopoeia are prepared by maceration method. Maceration is most widely used method of extraction. Maceration can be performed easily with pharmacist lab with small amount of sample. There are number of drugs which can extract by only maceration method due to their high swelling property or high mucilage content. So you see the maceration advantage. Now we are coming to the percolation. You might have seen this percolator, a cone shaped percolator. There are many shapes, but this is the cone shaped percolator where the menstruum can pass through the sample which you have kept in there. You can see the menstruum, the solvent system in the top, and washed sand which is kept there, and a filter paper, and the drug is kept in a layer, and down you can see glass wool. So we have to pack with the glass wool and the drug, and then keep a filter paper, then washed sand, and then you have to pour the menstruum and close it, and then you can remove the solvent system, or you can just continuously, even you can run the percolation. Percolation can be done. Continuous hot percolation is by means of Slit apparatus. This is by cold percolation is also done by means of cone shaped method. The word percolation refers to first passing through. Percolation can be defined as a short successive maceration or extraction by the method of displacement. Percolation can be performed into the conical percolator, cylindrical percolator, or steam jacketed percolator. So you see, there is the system can be easily drawn by any student. Menstruum, washed sand, below that you are keeping already that filter paper, then the drug layer, then the glass wool. So you have various stages. Size reduction, I am not given more explanation because the student maybe know why we are doing size reduction, how we are doing the size reduction, you may be knowing. We have discussed in the class about various methods of size reduction by cutting, compression, impact and attrition. These are the main principles which can be done for the size reduction or the comminuting for various things which can be done. Hard materials, dried materials have to be undergo size reduction so that increase the surface area so that the extraction can be improved by means of solvent can enter into the part where you want to solubilize in the drugs can be easily done. Imbibition means keeping in the menstruum or keeping in the solvent for the period of time. Soaking you can say, or you can keeping so that the constituents which is present in the crude drug can be solubilized by the menstruum or the solvent which you are using for the extraction process. Then come packing in this chamber for the percolator. After imbibition, after soaking, after keeping for the certain period with the solvent system or with the menstruum, after imbibition, lump the crude drug must be broken. Lump of the crude drug must be broken. The lower end of the percolator should be plugged with cotton and then place, or you can say cotton wool or the glass wool, that's what we have written, cotton. The drug powder layer is been kept as shown in the picture. The packing should be perfect, neither too tight nor too loose. Perfectly you have to do, which may affect the flow of the menstruum. If you are very tight, like a cement it is making, it is not possible for the menstruum to pass through and to make for the process of extraction. As we know that percolation means through, flow, first pass, it should pass through. 2/3 of the percolator should be covered with the drug, as shown in the picture, on which place the piece of the filter paper and washed sand can be kept above, should be placed at the top of the filter paper. This prevents any type of disturbance of the crude drug by the flow of menstruum. So now the fourth step in the percolation is maceration. Sufficient amount of menstruum should be added from the top as shown in the picture after packing the percolator. During the addition of menstruum, the low tap of the percolator should be open so that the air present in the percolator is displaced by the menstruum. The flow can be improved. When the menstruum starts to come through the lower tap, close the tap and allow to stand in the percolator for 24 hours as primarily maceration takes place. The menstruum level should be above the drug bed. And the last one, percolation taking place. Open the lower tap after 24 hours of maceration and collect the menstruum from the lower end until the 3/4 portion of the final product is obtained. Meanwhile, sufficient menstruum added over the powdered drug so that the pack does not become dry. Completion of the percolation process should be checked by various methods, like by checking the specific gravity of each few ml of the percolate which you have collected and compare with the specific gravity of the fresh menstruum. You can find out the difference. Evaporate the last few ml of the percolate to dryness and see that any residue comes after evaporation or not. Perform the specific chemical test for the constituents which you know. For the last few ml also of the percolate like tannins, glycosides, alkaloids, etc. Whether it is positive or negative. All the phytochemical investigation tests have to be performed by the investigator. And pressing the mark is also important in the percolation. In the last, the mark should be pressed and obtained liquid is added into the collected menstruum. More menstruum should be added to obtain the desired volume. Allow the liquid to stand and separate the suspended particles by filtration or decantation. Example, strong tincture of ginger, tincture of belladonna, these all can be prepared by this percolation method. Modified percolation. In modified percolation process, we reduce the drug percolate ratio 1:3, which is generally 1:4 in common percolation process. So we are reducing in the modified percolation. By this we can save menstruum time and heat. Percolation is type of displacement method. It has been observed that a stationary menstruum dissolve the more active constituents, because stationary menstruum for a longer period in the drug may remain in contact with more time to the drug compared to the moving menstruum. Now we want to discuss about the Slit apparatus, which we know, which is present in the lab. When you are coming to the lab, please, the students are more welcome to come and ask with me. Please show me because you remind me, when you come to the lab, I will show you the Slit. So when you see this vision or practicals are usually more important than a theory. Persons cannot, you will remember always in your lifetime. So you just see, I have not given a more explanation for this because I know it's possible for the students to write. This is Slit apparatus. You can see the original picture of that. The down we can see the heating mandle and the apparatus ground bottom flask which is fixed with the Slit, and above that you can see there is an water condenser. It's a series of Slit apparatus, 4 Slit apparatus in the picture. And you can see in the picture I have shown, the number 5 is the condenser which is showing there. And number 4 is the Solvent Extraction, the whole apparatus. And number 3, where the Slit himble, the apparatus, the part of the Slit apparatus, there is a shape which I have made there, the material will be kept. And you can see number 3, below the number 3 it is closed. You cannot, the solvent cannot come down, or the material cannot come down directly to the round bottom flask. And you will have a 6 number, there is a number 6, that is a siphon tube. Siphon tube, it is a bended tube. In the original picture also you can see a small one which is near to that side tube. The other one, 7th one and the 6th one is in between the 7th one in the original picture. But in the diagram I have shown the 6th here and 7th the opposite side. Both of them are in same side only, you can see there. Understand? So the small one is the 6th one, it is siphon tube, which brings the solvent back to the round bottom flask once it is filled in the 4th, the number 4, where in the diagram which I shown that Slit apparatus. So when you come to the lab, I can show you clearly and explain how it takes place. And even by means of showing you directly, the apparatus will give you more idea. But anyhow, here also you should know, you should write in the examination. You should write there is a round bottom flask and a Slit apparatus which contains siphon tube, and a side tube 7 and a siphon tube is 6, and the body is 3 is the place where the drug should be kept. And in the 5th one, which is shown, is the water condenser, which is again fixed in the Slit apparatus. So how it will operate? The down we can see the round bottom flask. The number 1 which I have shown is the solvent or the menstruum which is kept there. Here they have shown it is an ethanol which is a sample which they have taken. So and you can see the 8 number, that is an heating source. Usually they will keep heating mandle. So when you keep heating mandle, you can keep some temperature can be controlled for according to the boiling point of the solvent system which you are using. So once it the boil, once the vapor is formed from the round bottom flask, it will be taken through the side tube, that you can see the side tube number 7, and the vapor will come through the side tube and it will go up. It will not come down to the 3. It will go through the place where it shown the diagram 4 and it will go to the 5. So when it comes to the 5, what will happen? There is the water condenser. Simple water condenser. There is 2 inlet and outlet arrow they have shown. Cold water inlet and outlet will be their water. So what will happen? The condenser will be cool in a condition. So once the solvent system, the vapor enters into the condenser, what will happen? It will condense and drifts down and fall on the material which you have kept there. Where is the material? Where is the drug which you have kept? Crude drug? Number 3 place. So in the number place 3, the solvent which will be dripping down, the condensed one, vapor, fresh solvent, hot solvent will drip down. Slowly it will just go up and come to the stage even up to the 6th place, the number 6 which is on Siphon Tube. Can you see the picture? Watch the picture and so that you will understand. So that is Siphon Tube. So Siphon Tube means we can see even the liquid rises in the portion 3 as well as the liquid rises in the Siphon Tube also. It is like a bended tube, escape tube, like this. Small tube. And the liquid will rises both in the same time. In the solvent, in the 3 and also in the 6. So once it comes to the level of the 6 height and it drifts down, siphon, the name siphon, Pascal's law, it pulls down all the liquid down of the 3, the capacity, how much it is drips down. Because you know very well, the level when it is kept a bucket level in a little height and in the down, if you put a tube, fill the tube with the water and put it down, it will drain all the liquid which is kept in the side, in the top. In a tank, from a tank, if you want to remove the water, it is possible. If it doesn't have any tube connection, it is possible. Even how will you remove the water from a fish tank? How will you clean your fish tank in your home? If anybody is having fish tanks in their home, they may be knowing how the siphon will work according to the Pascal's law in physics. I'm not very sure. The Pascal's law and this is also having a small relation. That is different actually. Anyhow, it will siphon. So, this is continuous hot percolation method. Why? Because the liquid comes down back to the round bottom flask. And again, there is heat is generated from the bottom mandle, heating mandle. And the solvent will get evaporated and again it goes to the side tube and enter into the condenser, condense, fill in the area 3, come to the siphon tube, back to the... So this is continuous hot percolation method. If you do in 72 hours, continuously it will extract, very interesting extracts can be taken out from the constituents. So this is called continuous hot percolation method. So this is what we have seen here. I have not given explanation much and you have to go through it. An extraction of essential oil of the plant materials also comes to the different techniques, which is also an important one. Simple expression of pressure on a most citrus oil and Eculi method. This method is employed for citrus fruits which contain volatile principles. Here the oil contains ducts actually, which oil cells are ruptured by mechanical means or manually it is possible and it can be removed. That how you are taking, because we are not interested in the juice for our medicinal purpose. It is used for but we are in the skin lemon skin, orange skin, which contain hesperidin, which contain many medicinal activity constituents which are more interested. So we can use this type of mechanical means, squeezing in the points, needles, tubes, which can take the cells oil separated. So this is the purpose, special vessels taken container point projections. The raw material is twisted over the projections in clockwise direction and rupture the cells, oil comes out. This causes the oil cells, an essential oil, and the materials such that pigments also comes out and run through the center device. There will be channel, and so that it can be collected area. The liquid is thereby separated in the oil and removed from the water based part and mix, and it can be decanted. So this is a video. When you are seeing this, you will have an idea how they are manufacturing, and it was an interesting to show, and very interesting. For example, you can see here, these are oranges. Conveyor is used for taking these oranges. See, this orange skin contains oil. It's an interesting machine, how it operates. Just if you observe, you will be more interested to know about this action. Say, at the conveyor, it's just moving on, and there will be the pins which I have shown. The pins can prick the oil cells of the skin, and they can be removed. Just imagine that pins are rotating. That is polycitrus oil extractor. You see, now becomes, the skin will be totally removed by means of the pricking needle, pointing needles, and rotation of the conveyor. You see, it's slowly it's removing the skin of the oranges and taking the oil, orange oil. See that? Completely the skin have been removed. The oil extractor, you can see the pointer, pin pointer. This is lemon. In the same manner, lemon oil also can be prepared. In the same manner, the polycitrus extractor. And you can see the lemon also, oil emulsion prepared from both the lemon and orange separately, and then it can be compressed and the oil can be separated. Oil water emulsion is finished now. So oil completely separated from that water which may be present in that, and this is orange, that is lemon, and orange oil. How you can see that after taking that mechanical means also possible. But volatile oils are not very easy. This is orange oil, pure orange essential oil, and interesting to prepare. You see that, very costly. From the skin of the oranges it is possible for you to prepare. This is lemon, pure lemon essential oil is also prepared. So this is one type of amazing instrument which can be used for this preparation. And now there are many other methods are also they are following, like water distillation in extraction of essential oil, and the manufacture of essential oil using method, the water distillation, botanicals, the orange skin or lemon or eucalyptus completely immersed in the water and still, and it is boiled, and the method protects the oil so extracted to certain degree, since the surrounding water acts as a barrier to prevent it from overheating. When the condensed material cools down, the water and essential oil is separated and oil decanted and used as an essential oil. The water that is so separated is process is called used as a, even in the market they are not throwing that water. It can be used as a rose water, floral waters which is called hydrosol, sweet water, lavender water, rose water, orange water which is available. This is water distillation. This is the method. You can see this is an aroma oil, how they are preparing, keeping in the water and boiling, and then it will be separated. And you can see this is one type of same like Slit apparatus, but it is Clavenger's apparatus which is used for separating for volatile oil. Same like our round bottom flask connected with a bend tube, Clavenger apparatus. There is a water condenser, and the materials will be kept with the water and heating, and the volatile oil come to the condenser and condensed along with the water. If it is heavier than water, it will float in the water. If it is lighter than water, it is heavier than water, it will come to the down by gravitational force. If it is lighter than the water, if the volatile oil, then it will be floats, can be separated by the valve which is available in the Clavenger's apparatus. This is also an interesting apparatus like which is available in our lab. You can come and see. This is also an like substrate apparatus which is for small scale, which is properly. In large scale, you can use this type of apparatus in the laboratory by using vacuum, and even see water distillation can also be done by reducing the pressure under vacuum to reduce the temperature less than 100 degrees. It is beneficial for protecting the botanicals, and also thermolabile substances, essential oil can be separated. In large scale industry, they can be used this type of bigger instruments. The large instruments can also be used by means of using vacuum creation. And also you can see vacuum can be steam distillation. Another one, steam distillation for essential oil is also possible. When steam distillation is used for the manufacturing, instead of keeping with water the materials will be kept in perforated area. Steam will be passed through, which can take the volatile substance, and then it will be condensed. The botanical materials are placed in the still, and the steam is forced to pass through the material. That is the method. And this is hydro diffusion method. Materials which are kept in the top, and you can see micro oven from where it is drips down and comes to the bottom can be collected. When the material oil are extracted, hydro diffusion, this is another method in the type of steam distillation. The only varies the actual way of the steam introduced into the still. There from the bottom, which the steam is introduced from the still. But here the hydro diffusion of the steam fed from the top into the botanical material as shown in the picture. Instead of from the bottom, it will be from the top. So you can see it's also microwave type of extraction of volatile oils. Solvent extraction methods. When we talk about the broad term of solvent extraction, it does not only refers to the chemical solvent like hexane, but it also shows that solid oil, fat as well as carbon dioxide. Solvent extraction is particularly suitable for botanical material that has a very low yield of essential oil, or where it may be made up of mostly resinous compounds, and as such as delivers as far fingers fragrance that of distillation. During this type of extraction, non-volatile components of the botanical materials such as waxes, pigments are also extracted, or some cases this then removed during another process. Under solvent extraction, we can list as the following: maceration, enfleurage, and solvent extraction can also be done. So maceration method, with the maceration extraction method, the flowers are soaked in a hot oil to have their cells member rupture, and the hot oil then absorbs the essence. The oil is then cleared by botanicals and decanted, or you can say decanted, and the liquid and the volatile oil can be taken. This is much Same technique used for solvent extraction, where the solvent is used instead of hot oil used in maceration. Inflow-rich technique is a different technique, which you can say glass plates are kept with waxes, and the wax having the capacity to remove the volatile principle, and from the wax you can remove by means of alcohol, and that is the different technique, which is a very old technique, which is from the France they are started, and it is now also for the perfumery industry we are using this inflow-rich technique. Cold inflow-rich technique, hot inflow-rich techniques are also available. We'll just go through that in that very fast, you can see, and you can go through. And the glass plates are covered with a thin coating of especially prepared in an odorless fat, like that is called chassis. When you keep the wax in a thin layer in a glass plate, that is called chassis. And over the chassis you have to keep the petals of the flowers or the leaves of the volatile oil containing botanicals can be kept as shown in the picture. You can see the rose oil, roses petals will be kept over the glass plate which is having the wax, white color, that is called chassis, over which the petals are kept in the picture. The freshly cut flowers are individually laid on to it, and the fat which time becomes saturated with their essential oil, the fat will become saturated, it is absorbing the essential oil. The flowers are renewed, then you can remove the flowers, upside down you can keep, or you can, fresh flowers can also be kept. This is the chassis plates which have been used. Eventually the fragrance saturated fat known as pomades, the fat which is saturated with the flavor of essential oil, they are called pomades, and they can be treated with alcohol and oil can be extracted from the fat easily. So that is what inflow-rich. In cold inflow-rich, the large frame plates as shown in the picture, glass, they are called chassis, and smear thin layer of animal fat which is prepared, usually lard or the tallow or the pork or the beef respectively are allowed to set, and the flowers will be kept as shown in the diagram, and the botanicals, usually petals or whole flowers, are placed on the fat as shown in the picture and sent to the allowed to diffuse the fat over a period of 1 to 3 days, and then the fat will be removed and the chassis or the pomades can be treated with alcohol and you can remove the essential oil. The process is then repeated by replacing the spent botanicals with fresh ones until the fat has been reached to the desired degree of fragrance saturation. This procedure was developed in southern France in 19th century and production of very high grade concentrates. And in hot inflow-rich, so solid fats are heated and the botanicals are stirred over the fat. Spent botanicals can be repeatedly stained and heated, and the fat can be melted and replaced. The fresh can be also, we can replace again and again once it is saturated fragrance. This method is considered to be oldest known as procedure for preserving plant fragrance substances. And in the both instant cold and the fat saturated with the fragrance are called as inflow-rich pomades. This is how they used to use the remove, and they are the pomades. And the inflow-rich pomades are either sold as it was, and it can be used in hair cream, or it should be further washed and soaked, and it can be ethanol can be used to remove, and it can be used in the soap industry also, and molecules, and the alcohol is then, that alcohol is separated and the fat and allowed to evaporate, leaving behind the absolute of the botanical matter. The spent fat are used for the soaps, since it has some fragrance and it is very interesting for using. So this is hot percolation, hot inflow-rich technique, where the melts and the people are working over with the botanicals, infusions, maceration, hot inflow-rich techniques which they are following. Solvent extraction of essential oil can be extracted by using solvents with pure petroleum ether, methanol, ethanol or hexane, and it is often used as a fragile material. And such as jasmine, hyacinth, narcissus and tuberose, and would be able to handle the heat of the steam distillation. This is jasmine, hyacinth, narcissus and tuberose. These all are very common flowers which is available, and they are essential oil flavors which you are extracted. By means of Encourage technique, it is possible for you to do it. And now, when solvent extraction can also be done. But now we have to discuss about the solvent extraction. The solvent extraction are usually concentrated by means of using fragrant material, can also be used. And the main thing is we have to discuss about the hypercritical carbon dioxide extraction techniques, and which is one of the important techniques which also having an extraction. There are many methods, but still we have to go for this hypercritical carbon dioxide extraction. Is a fairly new way of extract essential oil from botanical materials, and although a bit of expensive side does yield good quality oil, carbon dioxide becomes hypercritical at 33 degrees Celsius. So, which is in a state in which there is triple state. It does not really gas or liquid, but has a quality of both, and it is an excellent solvent to use in extraction of essential oil. Since the low temperature required and the fact that the process is near to instantaneous, the carbon dioxide is further inert and therefore does not chemically interact with the essence, interact with the constituents which you are interested, that is being extracted. So to remove the carbon dioxide solvent, you simply need to remove the pressure under which it is kept. In normal room temperature and pressure, it can be removed. This process has taken place in a closed chamber for the hypercritical pressure required for carbon dioxide in 200 atmosphere. That is 200 times the pressure of the normal atmosphere. To achieve this type of pressure, have heavy duty stainless steel equipment is required, and this is where high capital, high investment is required for this extraction method. So this is just a schematic diagram where the carbon dioxide supply and a cooler and a pump will be kept, and you can see there is a oven where the extraction cell and a coil, preheated coil which supplies this carbon dioxide and liquid carbon dioxide, and then when the sample, you can see very easily it can be measured and it can be removed directly. The schematic diagram, but it is a very complicated instrument and expensive one. This is a picture which is shown about the critical point or supercritical point where the carbon dioxide, the pressure and temperature when you control, it can be a liquid and a gas in a stage. You can see in the physics, you might have stated the triple point, the critical point, where the carbon dioxide, the pressure and the temperature is controlled so that it can be in the form of liquid and a gaseous stage there, where the point, the triple point, which is arise, the solid, liquid and the gas states of any substances. Supercritical extraction in the phytochemistry, this property can be exploited for the maximum of extraction of the plant constituents. This is the industrial process. Supercritical fluid of carbon dioxide has been the environmental advantage over many common organic solvents and leaves no solvent residue in the product. There is no solvent residue. Solvent residue is the biggest issue for any type of extraction. So here, that advantage is there. It also allows a low temperature process and has proved the value for the extraction of reliable expensive fragrance of medicinal phytochemicals. So example of phytochemicals extracted by supercritical carbon dioxide alkaloids, decaffeination of green tea, fixed oils, volatile oils and resins. And in the coming classes, we will just go to the other type of extraction methods. We will continue, and even we will continue about the separation techniques and chromatographic techniques, and we have to go for various discussion. Thank you. I can continue, but still if it is more lengthy classes, students will be finding difficult to listen even in the internet, even I know. Make it short, usually they are telling, but our syllabus is wider, so I just made it. Thank you. Anyhow, just we'll see in the